Hello, this is our third experiment. It is called chemical aspect. It is, I think, chapter 3 in your books, where you actually studied about carbohydrates and proteins. Now, we call it chemical aspect or chemical molecules, or we call them as organic molecules or large molecules. Those three names, they're all for one, for organic compounds. Now, why do we call them organic? Now, we call them organic because they contain carbon in the building basic block. It's like a chain or a ring of a building block. It is made of carbon, oxygen, and hydrogen. And this is the, uh, the building block of organic molecules. Those organic molecules have different classification, 4 different classification. If I do have sugar attached to the basic carbon, carbohydrates, and oxygen, if sugar molecule is attached, I would classify it as carbohydrates. If it's one sugar molecule, we call it monosaccharide. If 2 sugar molecules are attached, we call it sucrose or maltose. If 3, polysaccharides, 3 and above. Now, to test for carbohydrates, we have 2 different tests. When we test for sugar, we use the reagent Benedict. And this is the definition of Benedict reagent. It's a blue colored reagent made of copper ions, basically 2 copper ions. If I add it to any solution that contains sugar and I reduce it, see the word reduction over here? Reduce, sorry. So if I reduce it by adding the test tubes into a hot water bath, not direct to a fire, hot water path, it will reduce from copper, from 2 ions of copper into 1. If sugar is present in the solution, it will change the color from the blue coloration of the reagent into a new color, okay, new chemical color. It is called brick red. And if you keep it 1 hour standing, it will precipitate in the bottom of the test tube. Now, for carbohydrates, for sugar, we have Benedict. For the simple sugar, that is monosaccharides. For polysaccharides, we have different reagent. It's called iodine, or the scientific name Ligules. But we call it iodine. Iodine is the one that we use it from pharmacies, the disinfectant one. I believe you know it also as the name of betadine. It's yellow browny color. When I add it to any substance or solution that contains polysaccharides, it will instant change in color from the yellow brown coloration to black blue. It reacts with the starch. Very weird, but yet it reacts with it. So we use it as a test to look for starch in food samples. As for proteins, now remember when I told you here, if sugar molecules is attached to the chain, to the chemical compound, carbohydrates, oxygen, and carbon, carbon, hydrogen, oxygen. If sugar molecule is attached to that, we call it as carbohydrates. The second classification is that when we add amino acid, not sugar, if amino acid is attached to a chain that contains hydrogen, carbon, oxygen, then it will be classified under proteins. Now, the test for protein, we use burette. So it's not Beirut, it's a burette reagent. It's a blue color, also made of copper. It reacts with the special bond of proteins. The proteins are bounded together or binded together with a very unique polypeptide chain. So it reacts with it. Also instant, we don't need to reduce, we don't need to heat, from the blue coloration of the burette into a purple or violet color. Instant color change, which I will show it to you. As for lipids, as you know, lipids, an example of lipids like oil, if you mix oil with alcohol, it will melt. It will bind together. So in order to remove oil from alcohol or benzene, we use something called Sudan red. Now Sudan red, it won't actually change any color. It actually binds to the molecules of oil, separates it from whatever soluble it is dissolved in. So the dye, this is Sudan red, will isolate lipids in a layer and color it in red. Very simple. So this is the procedure. You already have it. I will perform the experiment and you will have to fill that table with the results that I will show it to you in a minute. Answer these questions as an assignment and you will have to write a lab report there. Now the lab report, very simple. By now you have to learn about it. You have the result and you have the procedure and you have the information. Just summarize it and write your lab report. Okay? Now I will go for the practical part. And for any question, please do not hesitate. Through email, you can email it to me. So this is the performance of the experiment. This is the hot water bath I'm aiming for. This is a Clifton plate. It's a heater where I'm gonna heat the water to prepare my Benedict reagent. It's a reagent that tests for % of simple sugar, and I always use samples that contains sugar, and also I have to use water. It's written over here, it's labeled water. Now the reason I'm using water as a negative control, and I have to have a positive control. This is a glucose solution. This is the color that I'm aiming for, and as you can see, there is a precipitation. It's a beginning of a precipitation in the bottom of test tube. This is the color I'm looking for. Now why do I have to have a negative and positive control for accuracy and precision? I have to have an accurate and precise result. So to prepare that, just let me open it. Now 1 full withdraw of that pipette, actually it counts 1 milli, and I need to have 2 milli over here in each for you to see. I have to have a similar measurement of the sample and the reagent to have an accurate and precise result. So I'm just gonna use those test tubes over here. This is soda, sorry, this is juice, this is soda, and this is water. Soda, 7-Up. Oops, just a second, it went down. Here, this is much more easier. So I will have 2 full withdraw in order to have 2 milli of sample and 2 milli of the reagent. Now why do we have to have negative and positive control? For accuracy. Why do we have to have a very accurate measurement? For also accuracy and precision, to have an accurate and precise coloration. So very simple. Now there is nothing change over here, but I will add it to my hot water bath, and I will wait for 5 to 7 minutes for the target result I'm looking for. Now the precipitation over here, you won't see it immediately, you will see only the coloration, but if you keep it standing for 1 hour, you will see the precipitation. This is, I just do it before the experiment for you, just to see what you're looking at. This is a positive control. Now look over here. This is the negative control for Benedict. This is the soda juice. I can't hold it, but I will show it to you right now. And this is the juice. It's an apple juice, basically. I will compare it to my negative and positive. This is my positive control. This is my negative control. Now those two, when you keep it standing, it will precipitate in the bottom. This is the target color that I'm looking for. I will take a picture of it right now. Now, while we are waiting for our Benedict reagent results, I will perform my second result, اللي هو looking for the percents of starch and food samples. I have here my iodine, okay, my bread, my A4 size paper, the starch solution. This is just a precipitate of the starch solution, and my negative control. And this is my positive control. Okay, so look at the bread, look at the brownie coloration over here. It's a brown, yellowish, orangey, whatever you like to call it. Look, if I add the drop, how it changed into a black blue coloration. Look at the A4 size paper, because paper is made from plants. Plants has cellulose. Cellulose is polysaccharide, so this is definitely comes from plants. This is the water. Nothing happens to it. This is the starch. Instant color change. I'll just mix it. So this is my sample, this is my positive control, and this is my negative control. It's very important to compare my result to the positive and negative control for accuracy and precision. Okay? Beautiful, right? Our third experiment using Burette reagent for the percents of protein. I do have here whole milk, egg white, and my negative control, the water, and I will show you right now the positive control. This is my positive control. Look at the purple coloration. I will show it to you in a white background. Look, this is the target color I'm looking for. So 1 full withdraw is 1 milli. I need 2 because this is 2 milli over here. 1, 2, 1, 2. Instant color change. 1, 2. This is the milk. It's purple. Egg white. Negative control. Positive control. Perfect, right? Now, this is Sudan red. Okay? It has nothing to do with Sudan as a country. This is the name of the scientist, I believe, that called it Sudan. And this is a mixture of oil and alcohol, and this is water as a negative control. So I will add, as I said, Sudan red doesn't change any color, it only isolates the lipid molecules. Over here. So if I mix it, it's only, this is the negative control, this is the water. while look at the oil, it is isolated away from the alcohol. It's always on the top. Okay, it isolates it on the top. So this is how we know that there is oil in this mixture. We add Sudan Red, it captures or isolates all lipid layers on the top and keeping the liquid or solute in the bottom. Simple? Yes.